Suppose you're a scientist working on a vaccine and you're given a blood sample from which you need to extract the CD4 T cells. These T cells can then be used in a separate assay to determine antibody production, which tells us if the vaccine is working. First, let's look at the blood sample itself. Blood is composed of plasma, platelets, red blood cells, and white blood cells. There are 5 different types of white blood cells: neutrophils, eosinophils, monocytes, basophils, and lymphocytes. A CD4 T cell is a type of lymphocyte with a glycoprotein CD4 on its surface. So how would you go about separating such a specific cell? Well, one method would be through magnetic activated cell sorting. To prep for the cell sorting, a 50 nanometer microbead is conjugated to a specific antibody. This can be done 2 different ways, non-covalently or covalently. The 2 most common non-covalent methods are using a streptavidin-coated bead coupled to a biotinylated antibody and using a protein A or G to bind to IgG antibodies. The most common covalent attachments will form between an antibody and an amine group. The 4 most common magnetic bead coatings for covalent attachments are tosyl groups, hydrophobic beads, carboxylic acid hydrophilic beads, amine hydrophilic beads, and epoxy hydrophilic beads. Once that step is complete, you can introduce your blood sample, and the cell you want to separate out, your target cell, will bond to the antibody microbead coupling. The cell will have a surface receptor that will bind to the specific antibody. Now that our target cells have been magnetically labeled, we can run the sample through the magnetic field. This sample will run through a column filled with ferromagnetic steel spheres covered in a cell-friendly coating. These spheres will amplify the magnetic field by 10,000-fold. As it runs through, the microbeads, which are superparamagnetic, will attach to the steel spheres along with the antibody and target cell. The steel spheres are sized so that there is enough space between them for all the unwanted parts in the sample to pass through. Now the column may be removed from the magnetic field, and the beads, being superparamagnetic, will not retain any magnetic memory. So once removed, a buffer can be plunged through the column, and the microbead antibody target cell complex will be all that remains in the collection tube.